Hello everyone, my name is Davi. I'm a PhD student at Universidade de São Paulo in the postgraduate program in comparative biology. And today I'm going to talk a bit about my PhD project, which is entitled as Evolution of the Vocal Anatomy Associated with the Acoustic Diversity in Bokermannohyla. My advisor is Professor Flávio Bockmann, and my two co-advisors are Ariovaldo, Giaretta, and Thiago de Carvalho. Anurus are these sympathetic animals like frogs, toads, and tree frogs, and they communicate with each other through the emission of sounds and calls. Male individuals calls to attract females for reproduction, and therefore calls are susceptible to female preferences, since females must be able to recognize and select calls of its own species, and therefore calls tend to be unique among species. So, how calls are produced? It involves three anatomical structures like lungs, larynx, and the buccal cavity. Air that comes from the lungs passes through the larynx. Here is a half larynx. Within the larynx there are vocal cords that vibrate as long as air passes through it. So the sound produced by the vibration of the vocal cords within the larynx goes into the vocal sac, and the sound is then radiated throughout the surrounding environment, which in most cases is the air. So once calls tend to be unique among Anuran species, one might expect that different call structures might be the result of different anatomical modifications in the larynx. Basically, in our project, the objectives are to characterize the larynx calls in a correlated manner in a Brazilian genus of tree frogs named Bokermannohyla. Species in Bokermannohyla have calls that vary from whistle-like calls like this one to grunt-like calls like this one. So these are two examples, among others, of how call structures can vary within the genus. So the main question in my PhD project is whether differences or similarities in the larynx anatomy reflect changes or conservatives in calls of species of Bokermannohyla. So some of my preliminary results, like call characterization, we identified at least 5 acoustic patterns like this one. This is the first one. The second one. The third one. And the last one. So as you can see, there is a wide variation among species in terms of calls. About larynx characterization, and despite of that variation in calls, we actually evidenced that all species of Bokermannohyla actually share a basic laryngeal plan without any kind of anatomical modification that could explain such a difference in calls of species. So as you can see here, these are 3D representations of larynx of 5 different species, each of which are representatives of those different call patterns that I've shown previously. And as you can see, they share a basic laryngeal plan, albeit there are subtle differences like muscle width, which is here represented in colors, in the arytenoid cartilage, which is here represented in gray, and in the vocal cords within, you can see there are subtle difference in vocal cord width as well. So these results are actually contrary to what was first expected. The different calls among species could be explained by difference in the laryngeal anatomy. So we raised an hypothesis based on that different call structures actually stem from difference in how species control the larynx behaviorally to produce distinct calls instead of physical limitations imposed by the anatomy of the larynx per se. So for future directions, we will now extract morphometric measurements from the larynx as well as its associated muscle volumes. So we can seek now for potential evolutionary correlations between larynx and call traits through the application of phylogenetic comparative methods. So that's it. Thanks for watching. Feel free to contact me in these addresses if you have any questions. So thank you.